Okay, so it's really important that you follow the instructions carefully. So number 1 says label test tubes A to C. So we've done that, tube A, B and C. And it says using a syringe, measure 2 centimetre cube of protein solution into tube A. So it's important to remember to stir the protein solution first and have a syringe here. So 2 centimetre cube into tube A. Okay. And then using the same syringe, it says add 0.5 centimetre cube into B. So 0.5 into B. And 0 into C. So we're not actually putting anything into C. So we've put different volumes of the same protein solution into these tubes. What we're going to do now is add water so that we can bring all the volumes up to the same volume, but we will have created 3 different concentrations of protein. So point 4 says using a different syringe. So I've placed this syringe next to my protein solution. I know that was the syringe I used there. So a different syringe for the water. Now it says measure 2 cubic centimetres into tube C. So this is really important, like I say, to follow the instructions carefully, because it's telling us to work backwards now and actually put 2 cubic centimetres into tube C. So we're not starting with tube A. And then 1.5 into tube B. And it says 0 into tube A. So each tube now has a total volume of 2 cubic centimetres. C has 2 cubic centimetres of water and A has 2 cubic centimetres of protein. Now B, it has 2 cubic centimetres in total, but it has 0.5 of protein and 1.5 of water. So we have 3 different concentrations of protein made up. You know, point 6 says add 1 cubic centimetre of biuret to each tube. So if you remember, biuret was the test for protein, and it's a blue colour, as you can see, and if protein's present we should see this change to a lilac colour. Now remember, it's important to read the instructions fully before you actually start carrying out the procedure. It says here add 1 cubic centimetre of biuret to each tube, but then it says in point 7, starting with tube C, stir each tube using the glass rod, and immediately after stirring all tubes, record your observations in a table. So obviously you would have read this before carrying out the procedure, and you would have created your table before you actually put the biuret into these tubes, so that you can make that observation immediately, as it says here. Now that's really important. That suggests that the longer you leave the tubes, the more they'll change. And for the purposes of this experiment, they want you to record your immediate observations. So you might not actually be recording a correct trend if you leave it any longer. So you should create your table. You should do that now, so that you can pause the video and record your observations immediately after adding the biuret and stirring the tubes. So again, a different syringe here to avoid contamination, and we're adding 1 cubic centimetre into each. Okay, so 1 into A, 1 into B, and just need to take some more, 1 into C. So starting with tube C, and that's important, this is the one with the lowest concentration, so if we always stir from high to low concentration, we avoid contamination, but remember to wipe that glass rod in between. So you need to immediately record your observations. So if I just take these tubes and just place a white piece of paper behind them, you should be able to see them a little bit more clearly. So this is tube A, tube B and tube C. And you're recording your observations when you record your observations, think about 3 things. Think about what's the colour of the tube, think about whether if it's cloudy or clear, and can you make a comment on the comparison between other tubes. So if you're saying one tube is cloudy, is it cloudier than another one or less cloudy than another? You should state some sort of comparison with the other tubes. So record your observations in a suitable table.